We are going to talk about growth, aging, and cancer. Let's start with growth. Growth is considered as one of the most fundamental attributes of any living organism. It is defined as the increase in the organism's body size and mass with time. The growth of an organism is driven largely by cell division and also by secretion of the extracellular matrix. There are two factors influencing cell growth, that is, the rate of cell division and cell size. Growth involves an increase of cell number while the cell size is conserved such that the volume of a cell population is proportional to the cell number. There are two biochemical pathways of growth control. The first is the insulin system. It controls the overall size and controls the rate of protein synthesis. In the insulin pathway, insulin binds to its receptor in the plasma membrane and activates it through the transphosphorylation of downstream targets via adapter proteins, which activate two key signal pathways. The ERK pathway is activated through the phosphorylation of adapter proteins SHC, GRB2, and SOS. When an adapter protein called the insulin receptor substrate is transphosphorylated, it activates the PI3K or the PI3 kinase pathway. The second pathway is the Hippo pathway. It controls the organ size in animals through the regulation of cell proliferation and apoptosis. However, if the Yorkie is unphosphorylated and enters the nucleus, it leads to the overexpression of Yorkie, resulting to excessive tissue growth. Hippo activates another kinase called WARTS, and WARTS phosphorylates Yorkie, a nuclear protein that is retained in the cytoplasm when it is phosphorylated. Aging. Aging is considered developmental as it responds to genes that can be manipulated. One of the important systems controlling it is the insulin signaling pathway. Studies on mammalian cells grown in primary culture show that they tend to slow down and eventually stop growing after a certain number of passages. This is known as the Hayflick limit. There are two systematic processes that explain this. One is the INK4A or the telomere shortening. The telomere shortening is a mechanism that approximates to the ability of cells to count their divisions of many cycles. Cancer. Cancer is a type of inappropriate postnatal development. It is the growth of the body's own tissue to an inappropriate extent. Postnatal defects include hyperplasia and metaplasia. Hyperplasia is an excessive production but normal cell differentiation turnover. Metaplasia is the conversion of one tissue type to another. Tumors are an abnormal mass of tissue that result when cells divide more than they should do. There are two tumors, benign or malignant. Tumors are caused by somatic mutation, germline mutation, and gene expression changes. There are a couple types of histological cancer: dysplasia, sarcomas, carcinomas, leukemia, and lymphoma. Dysplasia is altered growth and differentiation within tissues or organs, which can lead to conditions that involve enlarged tissues or precancerous cells. Sarcomas are tumors in the connective tissues. Carcinomas are cancers of the epithelial tissues. Leukemia, cancers of the hematopoietic tissue. Lymphoma are solid overgrowths of lymphocytes that are accompanied by malignant cells. Cancer stem cells are a small subpopulation of cells within tumors with capabilities of self-renewal, differentiation, and thermogenicity. Cancer stem cells is the population that needs to be eradicated in therapy. When similar cells are grouped, they are called tissues. These group of cells come together to perform a common function. In a literal sense, tissue means woven. When tissues are combined, they become organs such as the liver, kidney, and the heart. The type of tissue defines its function. There are 4 primary tissues that each hold a different job from another. Epithelial tissue line our body cavities and organs and essentially cover and protect the body. Our connective tissues provide support, and the muscle tissue give us movement, while the nervous tissue provides us with control and communication. Epithelial tissues are a sheet of cells that are arranged on the basement membrane, with each cell joined to its neighbors by specialized junctions. Junctional complexes consist of 3 components: tight junctions that prevents leaking between the cells and isolates the components of the apical and basolateral membrane, adhesive junctions that are attachment points joining the microfilament networks of the cell, and desmosomes are the point of contact joining bundles of cytokeratin filaments. Connective tissues are dominated by fibroblasts, such as the dermis of the skin and the fibrous capsule surrounding most organs. Much of it is derived from the mesoderm of the embryo, although some is also formed by the neural crest. Fibroblasts are specialized to secrete the matrix components, which includes hyaluron, protoglycan, fibronectin, type 1 collagen, type 3 collagen, and elastin. There are 3 types of muscle tissue: skeletal muscle, smooth muscle, and cardiac muscle. Skeletal muscles develop from myotomes and multiply without differentiation. They are composed of elongated, multinucleated cells called myofibers. Smooth muscles is derived from the lateral plate of the embryo and is found mainly around the gut, blood vessels, and the duct of glands, where inherent rhythmic contraction is required. Cardiac muscles occur only in the heart, composed of cardiomyocytes. It has myofibrils, but like smooth muscle, it remains as individual cells. The cells are joined end-to-end by intercalated disc, which contains structural junctions, adherents, and desmosomes, together with gap junctions that allow rapid spread of electrical signals through the myocardium. The nervous tissues make up the nervous system. It consists of neurons and supporting cells called neuroglia. It is responsible for coordinating and controlling many body activities. It stimulates muscle contraction, creates an awareness of the environment, and plays a major role in emotions, memory, and reasoning. Nervous tissue also includes cells that do not transmit impulses but instead support the activities of the neurons. These are the glial cells, together termed as the neuroglia. Supporting or glia cells bind neurons together and insulate the neurons. Some are phagocytic and protect against bacterial invasion, while others provide nutrients by binding blood vessels to the neurons. The rate of cell division determines the renewal of tissue derived from rare stem cells. Cell replacement is the study of which makes up most of the postnatal developmental biology. There are many types and degrees of cell replacement. Some tissue types become largely postmitotic after birth or once the growth of an animal is complete. Others do not normally undergo cell renewal but have some regeneration capacity following damage, while others undergo continuous lifelong renewal. There are 2 misconceptions on tissue-specific stem cells. 1. It is often thought that the stem cells can turn into any cell type. 2nd, every tissue is often thought to have its own stem cells. Tissue types in the body can be classified on the basis of their proliferative behavior, but this classification only refers to mammals, as lower vertebrates may show uncommon patterns of cell renewal. There are 3 types. First, are tissues where the principal functional cell types are postmitotic, such as neurons, skeletal muscle fibers, and cardiomyocytes. and where they are mostly replaced when lost. Second, are tissues consisting of differentiated cells that grow by division while the animal is growing and stop when the adult is attained. And third, are renewal tissues such as the hematopoietic system, the epidermis of the skin, the epithelium of the gut, and the spermatogonia of the testis. The hematopoietic system brings about a hematopoietic stem cell that can both renew itself and also differentiate into a wide variety of cell types. In the adult mammal, this consists of organs and tissues primarily residing in the bone marrow, spleen, tonsils, and lymph nodes involved in the production of blood. Stem cells are the foundation cells for every organ and tissue in the body that is capable of unlimited division. This has two key properties. One is the ability to self-renew, dividing in a way that makes copies of themselves. Two is the ability to differentiate, giving rise to the mature types of cells that make up our organs and tissues. Tissue-specific stem cells or adult somatic stem cells are cells that continue to divide for the lifetime of the organism. They are committed to generate multiple organ-specific cell types and are found in several organs that need to continuously replenish themselves like the blood, skin, gut, and brain.
One example of stem cells are neoblasts. Neoblasts are planarian adult somatic stem cells that exhibit levels of plasticity and pluripotency comparable to embryonic and induced pluripotent stem cells. It includes pluripotent stem cells that sustain tissue homeostasis and regeneration of lost body parts in planarians. But before delving into planarian regeneration, let's first talk about regeneration as a whole. So, what is regeneration? Regeneration is the natural process of replacing or restoring damaged or missing cells, tissues, organs, and even entire body parts to full function in plants and animals. Among invertebrates, regeneration powers are especially more in sponges, cylinderates, flatworms, and tunicates. Some invertebrates can regenerate whole bodies from small tissue fragments. This is called whole body regeneration. Whole body regeneration is usually associated with a type of asexual reproduction in which the body can spontaneously fragment into two parts, and each develops to reconstitute a new individual. Planarians are the simplest animals that show bilateral symmetry and are capable of bidirectional regeneration. They are also known for their high regenerative capacity, wherein they can rapidly regenerate any part of their body. This occurs due to the abundance of stem cell-like cells or neoblasts in planarians, unlike other organisms. Now, let's look at how planarians regenerate. If cut in half, the anterior half will generate a new tail region from its posterior cut surface, while the posterior half will regenerate a new head region from its anterior cut surface. If it is cut lengthwise, the planaria will regenerate the missing halves. Following the transection, a wound epithelium forms across the cut surface. This results in a regeneration tissue called the blastema, which generally refers to a generation bud which contains undifferentiated proliferating cells. The blastema enlarges by cell recruitment and it redifferentiates to form the missing structures. On the other hand, Drosophila and other holometabolous insects are mostly postmitotic, thus no regeneration occurs. However, In hemimetabolous insects such as locusts or cockroaches, appendage regeneration occurs. If a leg or antenna is removed, it will grow again and will become visible at the next molt when the old cuticle is shed. Let's take a look at the process of insect limb regeneration. Immediately following amputation, the hemocytes of the blood forms a scab. Underneath the scab, epidermis then grows over the wound. The wound epidermis then forms a blastema and grows to form distal epidermal part. Complete regeneration takes place over 35 to 40 days or 4 to 6 molts. Among vertebrates, the power of regeneration is limited to the regeneration of limbs, jaws, gills, and tail. In vertebrates, only certain amphibian species can regenerate limbs after a surgical removal. Some of these species, like inuran tadpoles, regenerate limbs during the period of outgrowth and differentiation, but lose this ability as it undergoes metamorphosis. Other species, like Curodil, can regenerate all throughout its larval and adult life. Now, let's take a look at salamander limb regeneration. After the amputation, stump rapidly forms a wound epithelium by migration of epidermal cells over the cut surface. Then the internal tissues differentiate to a depth of about 1 mm from the cut. The cells from this region form a blastema consisting of loose packed mesenchymal cells surrounded by a thick epidermal jacket. The blastema will then proliferate for a while, forming a conical medium bud, thus calling it the cone stage. This will then proceed to the palate stage, where the structures of the limb redifferentiate in a proximal to distant sequence. The notch stage, or the early digit stage, is where the limb is nearly at the end of the regenerative process. The digits of the amphibian starts to form. Finally, the late digit stage, where the limb has been completely replaced. Complete regeneration usually takes 2 to 3 weeks for small larvae and months for large adults. The pattern of the regenerate conforms with that of the stump, such that all parts distal to the cut are replaced. Distal regeneration occurs even when the cut surface is proximal facing. This is known as the law of distal transformation, which holds for all vertebrate and arthropod appendages capable of regeneration. In the amphibian limb, it also holds for intercalary regeneration, which means regeneration of a tissue junction between parts that are not normally adjacent to one another. Since the limb is asymmetrical in 3 dimensions, it should, in principle, require 3 sets of regional codes to specify the pattern of differentiation in the 3 anatomical axes, namely proximodistal, anteroposterior, and dorsoventral. Among mammals, the only regeneration of significant external appendages is the regrowth of antlers in deer. However, there are some interesting phenomena that are usually called regeneration but are more properly referred to as hyperplastic growth. One of these is liver regeneration. An old question about regeneration is whether it is an ancestral character, which is a general property of living matter, or whether it represents a set of specific adaptations to the different circumstances faced by different types of animal. Evidence in favor of an ancestral character comes from the role of WNT and bone morphogenetic protein signaling in controlling the pattern of whole body regeneration in ancials. It is tentatively concluded that whole body regeneration is an ancestral character, although has been lost from most animal lineages. On the other hand, appendage regeneration is more likely to represent a derived character resulting from many specific adaptations.
Evolution and development. Before talking about derived characters, let us first talk about evolution. Evolution is the change in heritable traits of biological populations over successive generations. These traits are the expressions of genes that are passed on from parent to offspring during reproduction. Macroevolution refers to evolutionary patterns and processes above the species level. It encompasses the grandest trends and transformations in evolution, such as the origin of mammals and the radiation of flowering plants. The rise of molecular taxonomy has extended and refined our concepts of homology and analogy. Although many amino acids at inessential positions in a protein sequence will change because of neutral mutation, the amino acids at many other inessential positions remain the same. If a gene becomes duplicated, then only one copy is required to perform the original function, and the other copy is free to assume a different, although often related, function. In particular, among the vertebrates, a high proportion of genes belong to multi-gene families. Two genes in the same organisms that result from a gene duplication are called paralogs, while two genes in different organisms are called orthologs if they are directly related by lineage and preserve a common function. A phylogenetic tree is a diagram that represents evolutionary relationship among organisms. The construction of phylogenetic trees has two approaches. Numerical or phonetic taxonomy, wherein one involves scoring large numbers of characters, including continuous quantitative characters, and constructing the tree on the basis that more similar specimens should be closer together in descent. Next is cladistic or phylogenetic taxonomy, which involves applying certain assumptions about the likelihood of different types of change to try to deduce an actual phylogenetic tree. There is no guarantee that application of either method will produce the true tree, but the more data that are used, the more accurate the trees are likely to be. Zoologists have long drawn a distinction between two types of similarity between different organisms, namely homology and analogy. If two structures are homologous, it means not only that they look similar, but that this similarity is due to descent from a common ancestor, which possessed the ancestral version of the part in question. On the other hand, if two structures are analogous, it means that there is no common ancestry and the parts look similar because the pressure of natural selection has forced a convergence of structure to meet the need for a similar function. In general, characters that are thought to resemble of ancestors are called primitive or basal, while those thought to result from adaptive evolution are called derived. This figure shows a modern consensus phylogenetic tree for a few of the most important animal groups. Because of the lack of morphological characters in common between the adult animals of different phyla, information from embryos and larvae has traditionally been used to attempt to draw up the phylogenetic tree. The fossil record is often used to determine the phylogeny of groups containing hard body parts. It provides us a real view of the past, and it is the only source of time calibration for phylogenetic trees. The fossil record provides valuable time calibration for phylogenetic trees in the form of divergence dates, which are the points at which a new group appears for the first time. The totality of common expression domains has been called the zootype, to signify that this cryptic anatomy of developmental gene expression patterns defines what an animal actually is. One group of animal genes containing homeobox sequences is specifically referred to as Hox genes. This cluster of genes is responsible for determining the general body plan, such as the number of body segments of an animal, the number and placement of appendages, and animal head-tail directionality. Scientists have proposed that changes in a group of similar genes, or the Hox genes, have created the diverse animal body plans we have today.
That is all. We hope you enjoyed. Thank you for watching.